Dear friends, now we will move towards another method of recombination of selection of recombinants, that is blue-white screening. This blue-white screening strategy is used to discover or to find out the recombinants. As you all know that there is a lac operon which is having promoter, operator and structural genes that is ZYA. This Z particularly represents enzyme beta galactosidase. Scientists use this beta galactosidase enzyme for recombination because the beta galactosidase in presence of X-gal, that is a commercial product, X-gal, is converted into blue color complex. So it is a simple strategy to find out the recombinants, that is X-gal, which is converted into blue color due to presence of beta galactosidase enzyme. Scientists took this gene particularly from E. coli strain, that is M15, and near about 11 to 41 nucleotides from N terminal are deleted. So it is a recombinant, it is a mutant strain. That mutant Z gene is incorporated into vector. As you know that the vector is having property of antibiotic resistance, suppose ampicillin resistance property, and in this here they inserted mutant lac Z gene. So this vector is having ampicillin resistance gene as well as lac Z gene. Suppose we have this gene which is our gene of interest. This gene of interest or gene is get inserted into this, that is inside the lac Z gene. Why we take this? Because you all know that these genes is having the restriction sites. So if we use this gene and the gene of interest is get inserted inside the lac Z gene, suppose here we insert our gene of interest, then the lac Z gene will not intact. So no lac Z gene, because their property get lost, so it will not convert X-gal into blue color product. This reaction will not be possible, and the colonies appears. Suppose these colonies which are white, and the colonies which appear blue is having intact vector, that is having both ampicillin as well as lac Z gene. This screening is done particularly for identification of recombinant. So recombinant colonies are those colonies which shows white or white color colonies colony on plate ampicillin plate, that is what we call it as recombinant. And those which do not have the gene of interest will have the intact vector, and that intact vector will shows only blue colonies, these blue colonies. So blue colony indication there is no recombination. So if we screen particularly recombinant on ampicillin medium, then the colonies which having the white appearance are called as recombinants, while the colonies which shows the blue color is called as non-recombinant colonies. So in this way we can easily identify the recombinant with the help of the beta galactosidase strategy, that is what we call it as blue white screening. So thank you dear friends, these are some strategies. Beside these there are a lot of strategies which can useful for selection of the recombinants, but in our syllabus only two methods are incorporated, that is antibiotic resistance and blue white screening. So these strategies are useful for identification of the recombinants. More you can watch a lot of videos which is present in YouTube, as well as you will gather lot of information through various books. I will suggest you a book, that is David Freifield's Genetics, and that will be a good book for this. And also there is another book, that is Mole Bio by Upadhyay and Avinash Upadhyay sir. So thank you for this lecture. Thank you all.